manufacturer's restrictions when drawing therapeutic drug levels. CLSI also states that gel separator tubes should not be used for tricyclic antidepressants unless the gel tube manufacturer or your testing lab's internal studies establish stability. As long as red blood cells remain in contact with serum or plasma, significant and irreversible changes take place. Therefore, any delay in processing samples can change test results and impact the way a physician manages the patient. Some of these changes are accelerated by refrigeration. Although some analytes are stable in contact with the cells for up to 72 hours at room temperature, many analytes, including these, are not. To preserve stability, CLSI recommends that all tubes for routine chemistry testing be centrifuged and the serum or plasma removed from contact with the cells as soon as possible, unless evidence exists that longer contact times do not contribute to the inaccuracy of the results. Separation can be accomplished by collecting the sample into a gel separator tube or by physically removing the serum or plasma from the tube after centrifugation and transferring it to a properly labeled transfer tube with a fitted cap to prevent spilling. According to CLSI, once the serum or plasma is removed, it should be kept at room temperature no longer than 8 hours without refrigeration, and should be refrigerated no longer than 48 hours before freezing in order to preserve the concentration of unstable analytes. Occasionally, more serum or plasma is required than was originally recovered from a gel separator tube during initial centrifugation.
Sometimes those who process and centrifuge specimens are tempted to recentrifuge a gel tube. When we do that, we have to remember that when we recentrifuge a tube, we're combining serum or plasma that has been timely removed with that which has been allowed to remain in contact with the red cells. And it potentially yields a combination that's not likely to represent the patient's actual physiology. Studies have shown that recentrifuging specimens can result in a 47% increase in the patient's reported potassium level in those samples. So therefore, we have to make sure we're not recentrifuging specimens and that we're allowing specimens to clot fully to prevent the fibrin strands that can sometimes force us to consider recentrifugation. Proper clotting takes up to 30 minutes, so making sure we let our sample sit upright for 20 to 30 minutes and avoiding recentrifugation is critical to maintaining accurate test results. While we're on the subject of centrifugation, let's make sure we're not spinning our gel tubes with the stoppers removed. A study had shown that when gel tubes are spun with the stoppers removed, the potassium increases by 135%. So we have to make sure that we're leaving the stoppers intact when we spin our tubes that are going to be tested for potassium.
Many facilities separate serum from cells with plunger-type serum separators. Although such devices are a convenient way to accomplish separation, users should be cautioned about storing serum in these devices. Since they may not provide adequate separation from the cells, CLSI suggests that if you are using plunger-type serum separators, that an air barrier be created between the separator and the cells. In addition, make sure that the manufacturer has documentation demonstrating the absence of interference from contact with the device during storage. For complete blood counts, the literature suggests that EDTA tubes can be refrigerated up to 24 hours without affecting cell counts or morphology. However, since some individual parameters have demonstrated stability for up to a week at room temperature, refer to your tube manufacturer's recommendations for specific stabilities. Automated differentials are significantly less stable and vary with the laboratory's instrumentation. For sed rates, it is recommended that unrefrigerated EDTA samples be tested within 4 hours or within 12 hours if kept refrigerated. Reticulocyte counts drawn into EDTA tubes are considered stable for up to 6 hours at room temperature, 72 hours if kept refrigerated. Keep in mind that these are minimums and that some studies demonstrate longer stability. Sodium citrate tubes for coagulation studies can remain at room temperature after collection. However, it is recommended that PTTs be tested within 4 hours of collection. If the patient is heparinized, citrate tubes should be centrifuged and the plasma removed from the cells within 1 hour. Prothrombin times, however, are more forgiving and are stable for up to 24 hours at room temperature, even if uncentrifuged. Again, internal studies conducted by your testing facility may demonstrate longer stability. Bilirubin is especially sensitive to light and can deteriorate up to 50% after only 1 hour of exposure. To preserve the concentration of bilirubin in the neonatal samples you draw, protect them from light immediately after they're collected, and especially after centrifugation. And the light swapping tubes will work as in wrapping them in foil or encasing them in a light-tight container for transport to the lab. Occasionally, samples come out of the centrifuge with clotted serum. This is often caused by not allowing complete clotting to take place prior to centrifugation. Complete clotting can take up to 30 minutes. If you remove samples from the centrifuge and find yourself rimming clots, remember that you could be exposing yourself to bloodborne pathogens. You also risk hemolyzing the sample. Allowing samples adequate time to fully clot can prevent the necessity to rim clots. When samples are improperly collected or processed, patients can suffer devastating complications, even death. Additionally, it can cost hospitals hundreds of thousands of dollars in recollections, medication errors, and damages from legal settlements that can follow. Proper attention to detail and rigidly adhering to the standards for sample collection and handling is critical so that every patient is treated according to accurate results. Your role in patient care has never been more important. Thank you for watching, and remember, accurate results begin with you.